target identification is one of the most important steps of drug discovery. For this target identification, we have further to go through structural biology type things. Now, structural biology basically is a branch of biochemistry which provides us structural information about macromolecules. In two of the most important macromolecules are protein structures and their amino acids. Now what happens is that if we only have structure information about these macromolecules, then we will not be able to produce very good drugs. Why? Because in addition to structure information, we also require functional information. So structure information plus functional information of any macromolecule is important to understand its complete functionality and working. For that purpose, what we have done already, that there are many protein structures plus genome sequences available based on structural information only, but their functional information is missing, and that is a huge gap. So what are different methods for predicting functional information from the structural information? For that thing, number one, we should understand what are ligands and what are unligand parts of protein. So basically we are looking for only those parts which are ligand parts of the protein structure, which can be converted into medications or medicines. So for that purpose, what we usually use, we use techniques from structural biology such as X-ray crystallography and NMR spectroscopy to get information such as ligand information, active sites, protein-protein interaction sites from the given proteins. Another useful tool is basically protein databases, such as we have PDP, which is protein data bank, which provides us information that what is important inside the protein and what is not important, what would be used as a ligand and what would be unligand area. Similarly, we should have a complete information about the 3D structure. Why? Because based on that particular 3D structure, we can get functional information also. Since we can say that structure and function information, that can make two combinations, that either we have structure information and function information available, or structure is available, function is not available, and vice versa, that function is available but structure information is missing. So if we have got one thing, we can guess the other one. So what we have, molecular functions. So for defining molecular functions of some macromolecules, we can define those functions at two levels. Number one level is sequence-based methods. So sequence-based methods are those methods in which we have flat sequences of macromolecules. So flat sequences, for example, if we take example of DNA sequence, we will have only ACTG type things. If we take example of protein sequences, we have nucleic acid sequences, which are written in some flat file type thing. So what will happen? Whenever we try to work on these figures, they will be less accurate, but they can be calculated using classical bioinformatics tools such as multiple sequence alignment, and they can be used for finding important regions inside the protein sequences. So what are important sequences or what are the important regions in particularly in protein sequences? We can take examples of motifs and domains, so which were useful for converting some protein sequences into families and superfamilies or subfamilies. The other method is structure-based sequence or molecular functions. So what is the advantage of these things? They are based on the structure of the macromolecules. And then what will happen if we have structure of information for some macromolecule, then we can find inner details of that macromolecule as better as compared to the sequence information alone. So homology modeling, which was basically used for finding similarity between sequences, it can be a part of this structure-based approach. Identification of specific regions. Now what are those specific regions? Basically these are the specific regions which are useful inside the protein sequences. As I said earlier, those protein sequences are important part of protein sequences, can be motifs or our domains, and these motifs and domains, they are used for converting protein sequences into families, superfamilies, subfamilies, and so on. So where to start from basically, as we know that there are roughly 20 amino acids which have the specific location and position. So out of these amino acids, we have to decide which would be the locus or loci which will be used for ligand binding. Ligand is basically small drug-like molecule which binds with the protein to produce some medication. So again, we use MSA for structure and functional information. So what is MSA? Multiple sequence alignment, which aligns or assigns different sequences and tries to find out conserved regions in those areas. Now what is conserved region, or what is importance of conserved region here? So for that purpose, we can get information from two databases, which are, number one is PROSIT. PROSIT basically is a protein database which is used for finding out functions of newly discovered proteins. And how it discovers newly defined proteins? So it has list or details of previously defined proteins. Whenever a new protein comes, it tries to find out sequence alignment between the newly discovered protein and the already stored proteins. And if that there is some match or comparison is okay, then it will say that this is the part of the previous protein. Similarly, we have PFAM database. PFAM database basically, again, it assigns protein families to newly discovered proteins. But now comparison is not based on MSA alone. It is basically based on hidden Markov models. So hidden Markov models basically are used for aligning temporal based information or temporal based sequences, in which sequences are changing with respect to time. Then we have conserved regions related to families. Now what happens? What are the conserved regions? Conserved regions are basically those parts of proteins which do not change rapidly or which do not change over the time. So what is shown in this figure is that we have these conserved positions which are shown by this cross, and similarly here. And then we have these family dependent conserved position which are shown by this figure. So what happens is, we have shown here is that this active site basically, which is most importantly used for ligand binding or having some protein based interaction with another protein. So it can be seen that these conserved positions can be present at this active site also. So what we conclude from here is that conserved regions can be related to families. So if they are related to families, it would be very easy to identify unknown proteins when compared against the given or already developed proteins. Again, here we have given an example of conserved region of proteins. So basically we have different amino acids, and then we have shown structure of different organisms such as humans, which is shown by this first line. Then we have this mouse, then rat, cow, and then chimpanzee. So what we are showing here is that when we see these different things, such as if we say this part, so this is basically a conserved part which remains constant. Then again we have this next block, this is also constant, so this is also conserved. So whatever things which remain constant across organisms, we say that these are the conserved parts. Which have changed amino acid sequences, we say that they are not conservative or not conserved regions of proteins. So finally we reach towards how to find conserved regions in a protein sequence. For that way, or for that purpose, we have three different methods. Number one method is sequence space method. Basically, sequence space method is vectorial representation of multiple sequence alignment, in which different sequences are put in the form of vectors, and then we try to find out distances between different vectors. Then we have evolution trace method. This evolutionary trace method is basically can be understood by understanding phylogenetic tree level methods. So in phylogenetic trees level method, we have sequences which are sorted or which are arranged hierarchically. Hierarchically means that we have ancestors, then descendants, and so on. So grandparents, parents, and children, and so on. And finally we have S method, which is basically successive methods. So they are same as phylogenetic methods, but this hierarchical information is successive also, and information is informed or just applied at only one level, so we can get information about the protein sequence from single level also.